introduce yourself? Please state your name and any organization you're representing for the record.
My name is Jeff Chamberlain, and I'm a professor of neurology at the University of Washington. I'm also director of the Senator Paul D. Wellstone Muscular Dystrophy Research Center, and I'm a paid member of the Sarepta Scientific Advisory Board. I've been studying the molecular genetics of DMD for 30 years with a focus on dystrophin expression and the development of gene therapy. For these goals, my lab has developed transgenic mice. We've developed adenoviral vectors, lentiviral vectors, and AAV vectors in order to study how much dystrophin is needed to prevent or to reverse the pathophysiology of DMD. We've also been looking at the relative effects of producing full-length, Becker-like, and microdystrophin proteins in muscle. These studies have been remarkably consistent in showing that very low levels of dystrophin can have significant effects on muscle function. It was mentioned earlier that dystrophin levels as low as 10% of normal can prevent and largely reverse the dystrophic pathology, and our data in animal models certainly agrees with that. However, those levels are essentially what are needed for a cure, and we're not here today talking about a curative therapy. It's very important to emphasize that our studies in animal models also showed that much lower levels of dystrophin have a clear measurable impact on muscle function, and this is true whether we're expressing full-length dystrophin, Becker-like dystrophins, or even the microdystrophins that were developed in my laboratory. Our studies of dystrophin function have also demonstrated a mechanical role, mediating the lateral transmission of force from within a myofiber into the extracellular matrix. And the consequence of this is that a single dystrophin-positive myofiber has a clear protective effect on adjacent dystrophin-negative myofibers. Thus, the overall protection that's conferred by low dystrophin expression is greater than what you would predict by a simple comparison to normal dystrophin levels, and it's greater than you would see just by looking at the percent of dystrophin-positive fibers. We have clear data that even a single dystrophin-positive fiber protects adjacent fibers. So patchy or mosaic expression of dystrophin has a wider effect than just counting dystrophin-positive fibers. In fact, our studies indicate that any dystrophin expression has a beneficial effect on overall muscle function and physiology. In summary, our data in animal models acquired through a variety of different methods predict that the dystrophin expression patterns that have been observed with eteplirsen are sufficient to achieve a significant increase in muscle function. Thank you.